Hi, this video is about color oxidation. This is our group members. Color oxidation of meat reflects the meat's oxidative, antioxidative and reducing systems. Its discoloration shows the dominance of oxidative processes which is responsible affected by myoglobin concentration. Meat color is influenced by specific molecule at the binding site, the oxidation state of iron and the availability of concentration of oxygen. Myoglobin is predominant Haine protein and it exists in various redox forms such as deoxymyoglobin, oxymyoglobin, carboxymyoglobin and metmyoglobin. Deoxymyoglobin showed a purple-red color. Metmyoglobin provides a brown-red color. Oxymyoglobin and carboxymyoglobin gives a bright red color. Key factor for meat color protection is to maintain the appropriately reduced state of myoglobin. Vacuum packaging is the conventional physical way to protect meat color. It provides air barrier to inhibit the hypoxia-promoted formation of deoxymyoglobin. This experiment is to assess oxidative changes in meat color besides to compare packaging effects between vacuum and non-vacuum towards color stability and related quality attributes.
Expected outcome in color oxidation analysis. Expected outcome in color analysis using colorimeter. Expected outcome in pH analysis. Expected outcome in water holding capacity test.
Samples are divided into two types of packaging, which are sealed packaging and vacuum packaging, carried out at 3 different times: 0 hour, 3 hours, and 6 hours. The meat is cut and weighed before samples are sealed and vacuum packaged. The instrument is calibrated with a white ceramic plate. The sample surface is cleaned with a paper towel, and then the sample is cut to measure and record its color. For pH analysis, 5 g of meat is homogenized with 25 milliliters of distilled water using homogenizer. The pH electrode is inserted into the meat slurry and record the reading. Rinse the electrode thoroughly with distilled water. Place the meat sample at the center between 2 pieces of Whatman No. 1 filter papers. The filter papers with meat samples are placed on a flat surface and covered with plastic packaging. The sample is pressed using 2 kilograms of pressure or load for 2 minutes.
Weight of the pressed sample is weighed as W1.
Calculate water holding capacity using the following formula. The results of the analysis are shown in the table. Based on the result, there is a difference in color between meat in vacuum packaging and sealed packaging. Myoglobin is the water-soluble protein responsible for meat color. In vacuum packaging, the air is completely removed, creating a tight seal around the meat.
Without oxygen, myoglobin transforms into deoxymyoglobin. Deoxymyoglobin contains ferrous, Fe2+, iron with a vacant, no ligand attached, 6th coordination site. This gives the meat a darker, purplish color.
Meanwhile, sealed packaging, on the other hand, allows some air to remain inside. In sealed packaging, oxygen interacts with myoglobin, forming oxymyoglobin. This creates the bright red color. From the data, vacuum packaging slows down oxidation, helping the meat retain its natural color. However, over time, the oxygen in sealed packaging causes oxidation, and the oxymyoglobin breaks down. This leads to a loss of the bright red color.
Vacuum packaging can sometimes slow the rate of pH decline in meat. When meat is stored in a vacuum-sealed environment, the lack of oxygen can limit the growth of spoilage bacteria that are responsible for acid production. In non-vacuum packaging, the presence of oxygen supports the growth of aerobic microorganisms, including lactic acid bacteria. These bacteria can metabolize sugars and produce lactic acid, leading to a quicker decrease in the pH of the product.
Most of water in the muscle cell is held in the myofibrils. Meat in vacuum packaging has higher water holding capacity. Vacuum packaging involves removing air from the packaging. By eliminating air, vacuum packaging helps retain moisture within the sample. Compared to sealed packaging, the presence of air can lead to moisture evaporation over time and lead to greater water loss. Oxidation reactions in meat play a critical role in determining its quality, safety, and shelf life. These reactions primarily involve the oxidation of lipids, fat, and proteins within the meat, and their importance can be summarized. Lipid oxidation is a major factor in spoilage. The breakdown of fats into peroxides and secondary products reduces the meat's freshness, impacting its marketability. Myoglobin oxidation impacts the color of meat.